Well, thank you very much, Millie, for your excellent moderation. And thanks to Anne for all her very helpful comments on informed consent. So this brings us to the last session of the day. I guess the aim of this session is to showcase breakthroughs in medical science. And for this particular session, we've chosen two areas, mRNA vaccines and gene therapy. These are biological innovations with tremendous promise for a lot of diseases that previously were regarded as untreatable or difficult to manage. But they do raise unique considerations, not least of all for regulators. So I think we're all very excited to hear, first of all, from Karin Bok about mRNA vaccines, and she'll be followed by Nicole Verdun, who'll be telling us about innovative therapeutics and gene therapy. So, Karin, over to you.
Good afternoon. Thank you for attending this presentation. My name is Karin Bok. I am the Deputy Office Director of the Office of Vaccines Research and Review here at CBER FDA. And today I wanted to talk to you about mRNA vaccines. This is my disclaimer. My comments are informal and do not bind or obligate FDA, and I do not have any disclosures or conflicts of interest to declare. So at a glance, I wanted to give you a map of what the presentation will look like. We'll have a brief introduction to the topic. I'm going to tell you a little bit about mRNA vaccines technology, mRNA vaccines efficacy, which a lot of the data is from the SARS-CoV-2 vaccines, mRNA vaccines safety in general, also from vaccines we've been using for a while, like the SARS-CoV-2, and also biodistribution of mRNA in general. We're also going to talk about what we're still doing in terms of formula updates for vaccines, and how is the durability of the vaccines we're using via mRNA. Since the pandemic, there's been other approved mRNA vaccines, and we'll describe those. And of course, there's many of them that are in different stages of development. I'll tell you a few summary and conclusions that I think will be useful after listening to this. And of course, we have a couple of challenge questions for your entertainment. Okay, so we've been using vaccines for a couple of centuries now, but really the scientific advance on developing new vaccines and new technologies for vaccines has been in the last 70 years. Starting, of course, since we figured out how to grow viruses in vitro, and because of that, we were able to produce vaccines at larger scale. But since the discovery of the polio vaccine, we've made several advances. We figured out how to make recombinant vaccines like hepatitis B. We've had live attenuated vaccines like rotavirus. We have virus-like particles vaccines that have been very successful, like HPV. And of course, in 2020, we approved for the first time a new technology for vaccines based on mRNA. But even though mRNA is the new shiny object in our portfolio, and it's been very successful so far, we still have a lot of very advanced vaccine platforms and technologies that we're still using today. And we couldn't live without because different pathogens and different targets necessitate different platforms and different approaches. So we have inactivated viruses, we have viral-like particles and live attenuated viruses, and we have other kinds of platforms, or what we define as platforms, like viral vector-based vaccines that we also use not only in general but also for emergency situations. So what is the technology behind mRNA vaccines? So we've been able to figure out how to use mRNA inserted into a cell and translate a protein for a while, since the 90s. But there had to be several breakthroughs for us to come from that discovery all the way to today using mRNA as a tool for vaccination. There are several breakthroughs that are on this slide, and I'm going to describe to you. One of them was figuring out that we had to cap the mRNA. If you worked in a lab, you know that mRNA is very sensitive and it degrades very quickly. So capping helped with that. Tailing also helped with that, and also improving the translation efficiency of the gene of interest in an mRNA vaccine or molecule. We also use codon optimization in order to improve translation efficiency. And most importantly, we had to figure out how to get the mRNA molecule from the syringe all the way into your cells. And one of the most effective technologies to do that is through lipid nanoparticles. mRNA is also very reactogenic and can greatly stimulate the innate immune response. So scientists figure out that replacing the typical uridine of the RNA with pseudo-uridine, which is a modified molecule of uridine, reduces the reactogenicity by not stimulating the innate immune response so much. But even if you figure out all the RNA technology to have an mRNA vaccine, you still have the typical development and discovery issues that you have with any vaccine. You have to determine which one is going to be your antigen based on the pathogen that is your target. And if that antigen is going to be native and you're going to use it as is in the virus or in the bacteria, or you're going to modify it or add any other design to your vaccine. So this gives us a typical mRNA construct overview where you have your gene of interest in the middle. That's exactly what you want your mRNA to express, and that will be your antigen, your immunogen. But you also have to have an untranslated region in the 3'. You have to have a cap in the 5', poly A tail, a UTR, and all this, and of course like we said before, the pseudo-uridines instead of uridines. So all this enables the mRNA construct to be a successful vaccine. Manufacturing of mRNA vaccines is atypical from other vaccines we've seen so far, mostly because we consider this manufacturing technology to be synthetic, not use biological materials. We still use sometimes biological materials to develop the initial plasmid that we need in order to eventually synthesize the mRNA. But besides that, the whole process doesn't require biological materials like cell culture, fetal bovine serum, or any other biological material, and is done in a synthetic way. So you go from the sequence design to the in vitro transcription process. And then it's very important that you purify the unused reagents in your in vitro transcription process to have the pure mRNA molecule that is going to be your vaccine. And of course, then you have to have a method, a technology, in order to combine your mRNA vaccine with your lipid nanoparticle, and then filter the product before it gets formulated into a vial. So what happens once an individual gets injected with an mRNA vaccine? So mRNA vaccines can be transfected into muscle cells, and those are the cells that mostly translate the antigen of interest, but they can also be translated into antigen-presenting cells, which also is useful to activate the immune response. Of course, these antigens are transported to the lymph nodes, and they have lymph node resident cells that initiate the cascade of the immune response. But the muscle cells are the main recipients of your mRNA. So as we were saying before, it is important that you have modified uridines and you also have purified final mRNA product, because the innate immune response is very sensitive to any non-purified mRNA molecule, and you can have a very reactogenic vaccine, as you see here on the left, when a lot of the toll-like receptors and cytokines are activated when you inject an unpurified mRNA. And you, on the right, you see that if you use a well-constructed and purified vaccine, you let the adaptive immune response be the main consequence of that injection. mRNA vaccines have many advantages, like we discussed before. They are synthetic in manufacturing, they are faster to produce, they stimulate the innate immune response, but they also have other advantages because the antigen is translated inside the cell, meaning the antigen is delivered intracellularly. So they're not only able to produce antibodies and CD4 and stimulate the CD4 cells, they also stimulate CD8 cells. And that gives another branch of the immune response activated and ready to protect against an infection or disease. So the pros are that this is rapidly translated, vaccination is very fast, the RNA can be, of course, modified, and there is no prior immunity that can damp the immunization process, as in the case of other platforms like adenoviral vectors. You can sometimes have a vector where the individual that is being immunized has prior immunity, and that's going to lower the immunity level. And the last example we have here is a typical protein in adjuvant vaccine. While they're very useful for certain cases, the disadvantage of these approaches are that they don't have CD8 type immune response. So we have several overall advantages of mRNA technology for vaccines. Is the platform production, meaning that you can establish a method of production of the vaccine, and then by changing the gene of interest, you can use that same method of production for many different types of vaccines, which is not possible with other technologies. In general, it's biologics or cell-free production, which it's less complicated in terms of releasing or assays that you have to make sure you do in order to check the quality and the potency of that vaccine. There is a speed of manufacturing, again, because it is a synthetic and in vitro process, is much more is faster to scale and to manufacture in general. There is also faster speed of release because there are less assays required for release, just because of the cell-free production and biologic-free production. And in some cases, compared to some of other vaccines, the immune response might be more sophisticated, although this is not necessarily ideal for every pathogen you're going to have to work with. So this is just one of the advantages depending on the pathogen of interest. So what is the efficacy of mRNA vaccines? So during the pandemic, the US had an Operation Warp Speed, to develop many types of vaccines. They attempted to develop 2 mRNA vaccines from Moderna and Pfizer. And those vaccines not only were mRNA-based, they also had a specially designed antigen that was delivered intracellularly. So both of those approaches contribute to the final efficacy of a vaccine. But initially, when the original SARS-CoV-2 virus was circulating during 2020, and we were able to study in a large phase 3 trial the efficacy of these vaccines, mRNA vaccines proved to be very efficacious. In this case, you have both Pfizer and Moderna's initial results, and you can see that both are more than 90% efficacious, and even more for severe disease. and this was considered a huge success. It still will be considered a huge success for any vaccine that we study. And for instance, compared to the adenovirus-based vaccine, that was a different platform also being developed at the same time, which was slightly less efficacious with 72%. And not only that, you can see in these Kaplan curves how really the control and vaccinated groups are separated early, even a little bit over a week after vaccination. And it's very clear that vaccination is very, very effective against disease, and especially, like I said before, against severe disease. And in the case of the vector-based vaccine, the efficacy was a little bit lower, and the control group was not as separated from the vaccination. But overall, any of these results, 90% efficacy, 70% efficacy, it's considered a very successful vaccination. So we also continue to study vaccine efficacy and effectiveness, not only during the original approval of mRNA vaccines, but eventually when we had boosters, and we were able to observe that if a booster is well matched with the circulating virus, the vaccine continues to be very efficacious at preventing disease. We have, as I said, other vaccines in the market. The first one is a RSV-based mRNA vaccine, and this vaccine has also proven in a large phase 3 trial, and eventually was approved with this trial as a base of evidence that was also very effective at preventing RSV disease, and the efficacy is also over 90%. So in general, not only talking about the trials that were used to approve or authorize these vaccines, we also followed vaccine effectiveness once the vaccine was used in the US population. And as you can see here, the difference between the rates of disease in vaccinated and unvaccinated is really outstanding, especially during the early 20 or late 2021 when the virus was circulating. So these vaccines are very effective and have been proven to be very useful in the field as well, not only in the trials. So since we know that vaccines are really being effective in the field, I wanted to also talk about the other parts that we study when we evaluate a vaccine, which is safety, and to evaluate the benefit risk ratio of a vaccine. So what is, first of all, the biodistribution of mRNA once it gets injected into the arm? Of course, it's important to highlight, even though I'm sure you all know this, that mRNA does not travel to the cell nucleus as opposed to DNA. mRNA is biologically always present in the cytoplasm, and it doesn't travel to the nucleus, which is why it's impossible to get incorporated into our DNA. The antigen, however, can be detected in blood as soon as 24 hours after vaccination, and but it's rarely detected after the second dose of vaccinations. Some studies have shown that the antigen, in this case the spike for SARS-CoV-2, is also detectable in germinal centers in the lymph node for a long time, 120 days post-vaccination. So while you might not be able to detect it in blood, it is usual for antigens to stay for a long time in germinal centers. mRNA, in general, the vaccine itself is found in blood within hours and for approximately a month after vaccination. And the mRNA LNP, which is the technology we use to get the mRNA inside the cell, is detected at the injection site and is processed by the liver. So it's also detected by the liver. And LNPs, both LNPs, mRNAs, and also the antigen can be detected in various organs and sometimes in breast milk or other tissues. And the biodistribution in general seems to be correlated with different types of LNP formulation. So safety has always been a priority when developing any vaccine, and it was certainly a priority in the middle of a pandemic when developing the SARS-CoV-2 vaccines. Although we, as you can see on this slide, we took only 11 months to authorize, develop and authorize a COVID-19 vaccine compared to other vaccines that took years and years to be designed, developed, and approved. Safety was still a priority, and it was not compromised in the development of the vaccine. And why can I say this? Because even though it was less than a year to develop that vaccine, the work that contributed to the development of that vaccine had started at least 10 years before. And the development of the vaccine was done not only by one sponsor, but by in this case for the Moderna vaccine, by a group of very experienced scientists and vaccinologists that contributed to the speed of development of that vaccine. But the work that had been happening 10 years at least before the vaccine started to be developed was the work that helped us understand what kind of antigen we needed to use, how the antigen was going to be modified to be most effective, and how we could incorporate it into an mRNA vaccine and have a successful vaccination. So all that work had been already been done with other coronaviruses and with other viruses. So we were very well prepared for the very, very rapid development of the SARS-CoV-2 vaccine. And safety was also a priority because we know that there are cases, especially with respiratory viruses, of vaccines that have been designed in a very basic way, and they can cause what we call vaccine-enhanced disease. So we did have examples of very basic vaccines, in this case, formaldehyde inactivated vaccine that had a severe safety signal. So we were very careful, and we had all the scientific tools and knowledge in order to try to ensure as much as we could the safety of that vaccine. So we took, with the United States governments took risks developing those vaccines fast. Mostly those risks were financial. We did a lot of things at the same time without knowing whether the vaccine was going to be successful, especially manufacturing a lot of vaccine without knowing if that vaccine had even been successful in a phase 3 trial. But we did not take safety risks or other scientific risks. We use standard phase 1 and phase 3 protocols, and the vaccine follow-up on safety was ongoing. It was a little bit shorter by the time it was authorized, but it kept being monitored after that. So what is the benefit risk analysis of a vaccine, and how do we think about this in the case of mRNA? So there are certain safety events that have been described for the mRNA SARS-CoV-2 vaccine for both Pfizer and Moderna, and in this case is myocarditis and pericarditis is one of those examples. And you can see here that there is some elevated risk after vaccination, especially before 10 days after vaccination of pericarditis and myocarditis. And yes, every intervention, every vaccine, every drug is going to have some risk, and that's why we always assess the benefit over the risk of vaccinating an individual. But we carefully understand and survey any kind of safety event, in this case myocarditis and pericarditis, and inform the public of those safety events. It's also important to keep in mind, especially during this benefit risk analysis, that some of the safety events are also part of the original infection, the virus infection. And as you can see on this CDC warning, that the heart complications are higher after COVID infection than after any mRNA vaccination. So you always have to keep in mind both what are the benefits and what are the risks of vaccination. And that's what we do at FDA. So what is mRNA has been described as maybe not as durable as other platforms. So what is the mRNA vaccine durability, and how do we work to have formula updates? So is the immunity to mRNA express antigens durable? I think that question depends both on the mRNA vaccine and also on the kind of antigen you're trying to insert. If the natural infection is not durable, it's very unlikely that the vaccine durability is going to be more durable than the natural infection. So we know that mRNA vaccines produce antibodies that last about 6 months. As you can see, it starts declining at lower levels at 6 months. We also know that we have memory cells and that we will likely have to rely on those memory cells and memory T cells to recall the immune response if we get re-exposed to the virus. But in terms of antibodies, yes, the immune response declines, and you will likely have to be either reinfected or revaccinated in order to increase that antibody immune response, but you still have memory B cells and memory T cells that are able to be recalled. So how we approach updating vaccine composition, this is a similar process as we do with influenza vaccines. We backtrack the vaccine campaign and how much time we need to give manufacturers and sponsors in order to effectively manufacture the selected strain or the selected new booster. And we also have a clear list of requirements. What is the CMC data, the non-clinical data, and the immunogenicity data that they will need to provide in order for a new booster vaccine to be approved once the advisory committee makes the recommendation on the composition decision. So all those campaigns are followed every year and optimized every year. And we anticipate that this process, the process we use for influenza, will also apply in general to COVID-19 vaccines moving forward. So what is our role in general for COVID vaccines, including mRNA vaccines? We do have an advisory committee that does strain selection, and we have reference standard production. We do work to have lot release ready for seasonal vaccination. We evaluate safety and efficacy, and we help with post-market surveillance and advancing vaccine technology. And one of the most important things we do is helping to ensure public confidence by being very thorough when evaluating safety and efficacy and analyzing the benefit-risk ratio. So the last thing I wanted to talk to you about is the future of mRNA vaccines. We have approved mRNA vaccines like SARS-CoV-2 and RSV, but we have many others that are being developed. So we have examples for cytomegalovirus. That's one that is pretty advanced. We are close to also the end of development for influenza virus mRNA vaccines, HIV, Zika, rabies, and VZV, among others. This is just a summary example of all the, in this case, viral vaccines that are being developed with mRNA technology. So my summary and conclusions are mRNA technology was being developed for decades before the emergence of SARS-CoV-2. That's why we were able to use it so fast and so successfully. mRNA is an effective method to deliver an antigen. Careful vaccine design and antigen consideration is still needed, like for any vaccine. mRNA vaccines have been demonstrated to be safe and effective against COVID-19. and many other mRNA vaccines are in the pipeline, including the recently approved RSV mRNA vaccine. The ability of the mRNA platform to induce durable immunity is incompletely understood and still being studied. The mRNA platform advantages and cell-free manufacturing make it an ideal system for not only rapid response against emergencies, but also, of course, for the yearly updates of COVID-19 vaccines. So my challenge questions are, mRNA vaccines are the newest and most effective vaccines on the market. Do you think that statement is true? It is not. There are many, many technologies that are effective, and they have different advantages and disadvantages that might work better for your design depending on your target pathogen. mRNA vaccines have the advantage of eliciting CD8 positive immune responses through intracellular delivery of the target antigen. This is true. This is one of the advantages of delivering an immunogen intracellularly, just like the virus itself does it and other platforms do it. So having another part of the immune system stimulated is also helpful. Thank you very much. I hope you enjoyed the presentation, and looking forward to the questions. Bye.